Chapter number 3: The Modern Understanding of Scoliosis as a Whole Body Condition. The past few decades have seen a profound shift in how scoliosis is understood and treated. Once viewed solely as a mechanical problem of the spine, scoliosis is now increasingly recognized as a whole body condition, influenced by a complex interplay of genetic, neurological, hormonal, and biochemical factors. This emerging perspective, backed by advances in genetic testing, neurotransmitter research, and hormone analysis, is reshaping treatment approaches and offering hope for more personalized, effective interventions. From curve to complexity. Historically, scoliosis management focused almost entirely on the physical curvature of the spine. Treatments such as bracing and spinal fusion surgeries were designed to straighten the vertebral column, often without addressing the underlying causes of the deformity. However, clinicians and researchers began noticing that mechanical correction alone did not always halt curve progression, improve function, or prevent associated symptoms like chronic pain, fatigue, and postural instability. As research expanded into genetics, neurology, endocrinology, and molecular biology, it became evident that scoliosis is not merely a spinal issue. Instead, it represents a complex, multi-system condition involving dysfunctions across the body's biological networks. This realization prompted a more holistic view of scoliosis, one that sees the spinal curvature as a symptom, not the root cause. Genetic variant testing, unlocking hidden influences. One of the most groundbreaking developments in the modern understanding of scoliosis is the application of genetic variant testing. Through genome-wide association studies, GWAS, and targeted single nucleotide polymorphism, SNP testing, researchers have identified specific genetic markers associated with scoliosis risk and progression. Studies have revealed that adolescent idiopathic scoliosis, AIS, the most common form, has a significant hereditary component. Variants in genes involved in connective tissue integrity, such as FBN1 and COL11A1, bone growth and mineralization, VDR, PTHLH, neurotransmitter metabolism, COMT-MAOA, and hormonal regulation, ESR1, POMC, have all been linked to scoliosis susceptibility. Genetic testing can now uncover predispositions to weakened bone structure, impaired collagen synthesis, abnormal spinal growth patterns, dysregulation in neurotransmitter and hormone pathways. Importantly, many of these gene variants do not directly cause scoliosis, but instead create a biological environment that increases vulnerability when combined with environmental or developmental triggers. Genetic insights enable early identification of high-risk individuals and open the door to preventative strategies rather than reactive treatments. Neurotransmitter research, the brain-body connection. Another major advancement in scoliosis science involves the role of neurotransmitters, the chemical messengers that regulate communication between nerve cells. Several studies have shown that individuals with scoliosis often exhibit abnormalities in neurotransmitter systems, particularly those involving serotonin, dopamine, norepinephrine, and gamma-aminobutyric acid, GABA. For example, serotonin dysregulation has been associated with impaired postural control and balance deficits in scoliosis patients. Dopamine pathway dysfunctions are implicated in abnormal proprioception and motor coordination. GABA signaling imbalances may reduce spinal stability by affecting muscle tone and reflex integration. Research suggests that scoliosis may, in part, arise from faulty neural processing of postural information. In this model, the brain misinterprets sensory input about body positioning, resulting in maladaptive muscular responses that eventually lead to spinal asymmetry. Testing neurotransmitter levels through urinary, salivary, or blood assays can reveal imbalances that contribute to curve progression or treatment resistance. Interventions targeting neurotransmitter health, such as dietary changes, supplementation, neuroplasticity exercises, and functional neurology therapies, are becoming increasingly integrated into scoliosis care protocols. Hormonal research, growth, and curve progression. Hormonal factors are also now recognized as critical players in scoliosis development, especially during adolescence, a period marked by rapid hormonal shifts. Several hormones have been implicated in scoliosis onset and progression. Melatonin. Early studies found that animals deprived of melatonin developed scoliosis-like curves, leading researchers to investigate melatonin dysfunction in humans. Many scoliosis patients show disrupted melatonin rhythms, affecting growth regulation and bone health. Estrogen. Estrogen influences bone growth, ligament laxity, and musculoskeletal integrity. Polymorphisms in estrogen receptor genes, e.g., ESR1, have been linked to scoliosis risk and curve severity, especially in females. Growth hormone, GH, and insulin-like growth factor 1, IGF-1. Excessive spinal growth relative to surrounding muscle development during puberty has been proposed as a mechanism for scoliosis. Imbalances in GH and IGF-1 signaling may exacerbate this discrepancy. Progesterone and cortisol. Imbalances in these hormones can contribute to ligamentous instability, impaired tissue repair, and systemic inflammation, all of which may influence scoliosis progression. Modern hormone panels, available through specialized laboratories, can assess these hormonal pathways. Addressing hormonal imbalances early through functional endocrinology strategies may reduce the risk of curve progression and improve outcomes when combined with physical rehabilitation. A new era of personalized, whole-body treatment. The recognition of scoliosis as a whole-body condition has sparked a new paradigm in treatment, one that goes beyond bracing or surgery to encompass genetic, neurological, hormonal, and lifestyle interventions. Modern scoliosis management may include genetic testing, identifying high-risk gene variants to guide proactive monitoring and early intervention. Neurotransmitter support, implementing targeted nutritional strategies, neurofeedback, and functional neurology exercises to improve brain-body communication. Hormonal balancing, correcting melatonin, estrogen, progesterone, and cortisol imbalances to support healthy growth and tissue repair. Nutritional optimization, ensuring adequate intake of bone and nerve-supporting nutrients, such as vitamin D, magnesium, and B vitamins. Musculoskeletal rehabilitation, using specialized exercise programs such as scoliosis-specific rehabilitation or boot camp models that retrain posture, balance, and spinal stability. Environmental and lifestyle modifications, addressing sleep, stress, physical activity, and toxin exposure to optimize systemic health. This integrative, systems-based approach aims not just to straighten the spine, but to optimize the biological environment so that the body can support spinal health from within. The modern understanding of scoliosis as a whole-body condition marks a radical departure from the narrow mechanical models of the past. Advances in genetic variant testing, neurotransmitter research, and hormone analysis have illuminated the complex, interconnected systems underlying scoliosis development and progression. Rather than seeing the spinal curve as the disease itself, today's clinicians increasingly view it as a symptom of broader biological dysregulation. This shift has paved the way for more sophisticated, personalized, and effective interventions, treatments that offer patients the possibility not just of straighter spines, but of healthier, more resilient lives. The story of scoliosis is no longer just about bones and braces. It is about biology, balance, and the body's remarkable capacity for adaptation and healing when given the right tools and support.